Okay, well, good morning, everyone. I'm Vincenza Cifarelli, assistant professor in pharmacology and physiology at St. Louis University School of Medicine. First of all, I would like to thank the FDRS for organizing this great meeting and for the opportunity to speak today. I have nothing to disclose. So we all agree that a key feature in lipedema is adipose tissue expansion, which has been studied for a long time in the field of obesity. And there is an agreement that adipose tissue expansion is characterized by key steps. And so let's review them. So it is generally accepted that during nutrient excess and expansion of fat mass, there is an activation of adipocytes that start producing pro-inflammatory cytokines and chemokines, and that activates adhesion molecules on the surface of your vasculature that recruit circulating monocytes inside the adipose tissue. And so these infiltrating monocytes are quite dangerous because they will differentiate into macrophages, continuing to produce pro-inflammatory cytokines, chemokines, but also some markers of collagen deposition and fibrosis. So it is also generally accepted that as you progress towards these steps, the adipose tissue loses the insulin sensitivity, and together with a level of inflammation and level of fibrosis, you develop adipose tissue insulin resistance. Now, using different preclinical models of disease, it has been shown that once the adipose tissue becomes really fibrotic and stiff and cannot expand any longer, and so the fat, it started to be sent in the circulation together with inflammation targeting distant organs. So that is when you have higher chances of developing insulin resistance in type 2 diabetes or atherosclerosis. However, the model that I've just described, it's just a model. The clinics is much more complex, and there might be some exceptions to this model. And in fact, it is very well reported in the literature that there are some people that, despite having the same amount of fat, they are protected from developing metabolic abnormalities. They are not going to become insulin resistant. They have very low risk of developing type 2 diabetes, and they have very low risk of cardiovascular disease. So we do not understand why some people are protected from the detrimental effect of accumulation of adipose tissue, and others instead are more predisposed to become type 2 diabetic. And so when I was in the Center for Human Nutrition at Washington University School of Medicine, together with Samuel Klein, who is a co-investigator in this project, we wondered whether or not women with lipedema are metabolically healthy or metabolically unhealthy. And so what are the mechanisms of lipedema adipose tissue expansion? And so in order to answer these questions, we conducted a comprehensive studies that looked at whole body, organ system, and cellular, metabolic, and immune cell characterization in people with obesity and lipedema, and we matched them with controls without lipedema. We also conducted a study where we evaluated the effect of diet-induced weight loss on women with lipedema. And so the main question was to determine whether weight loss has the same beneficial effect on adipose tissue remodeling and metabolic function that is usually reported in classical obesity. So our study participants were recruited from the clinic of Dr. Thomas Wright, and after a screening visit, they underwent baseline testing that included body composition, which we assessed by DEXA scan. Those are X-ray scans, and they're able to determine total body and leg fat masses, the android to ganoid fat ratio, and whole body fat-free mass. We also employed MRI to measure the content of thigh and abdominal subcutaneous adipose tissue, the intra-abdominal adipose tissue volumes, and intrahepatic triglycerides. The metabolic function was measured using oral glucose tolerance test, as well as hyperinsulinic euglycemic clamp, which are state-of-the-art techniques to determine whether your organs, such as liver, skeletal muscle, and adipose tissue, are still insulin sensitive. And those are state-of-the-art techniques to do those measurements. We also look at adipose tissue biology by looking at the number of the content of immune cells, as well as gene expression by using RNA sequencing. So after baseline testing was completed, which usually takes about 2 to 3 weeks, all study participants initiated this diet-induced weight loss that was supervised by a study by a nutritionist as well as a behavioral psychologist that met with the study subject every week. Usually this phase lasts 4 months because we really want to have a progressive and steady decrease of the weight loss. And once the goal was reached, we repeated testing as shown at baseline. So this is our study subject flow. We were able to enroll 41 women in the study. For the obese LIP study, we enrolled 17 subjects, but only 11 were able to complete testing. Some of them dropped out from the study for several reasons, and 8 of them were able to complete testing before and after weight loss. As you can see, we also have a lean group that was included to have, to compare obesity and lipedema and obese only versus lean. So the obese and obese LIP groups were purposely matched on age, sex, race, BMI, fasting glucose, and 3-hour OGTT plasma glucose. And so when we look at body composition, so as we expected, we saw that the android to ganoid ratio is lower in women with lipedema as compared to the obese group. And this is mainly due by the fact that there is an increased leg fat mass and percentage, as well as an increased thigh subcutaneous adipose tissue volume and percentage. However, to our surprise, the content of liver fat between obese and obese LIP group was actually similar. When we look at the metabolic characteristics of the study subjects, as you can see, there was not much of a difference in terms of blood pressure, lipid content, or cardiometabolic parameters between lipedema and obese patients, with the exception that the glucose RD highlighted in red is increased in the lipedema group, meaning that that is the glucose rate of disposal, meaning that the insulin sensitivity in the skeletal muscle is way higher in the lipedema subject. So in theory, this patient population should be protected from developing cardiovascular disease. And this was another very surprising result. We also look at plasma markers of inflammations. And again, we couldn't really see much of a difference between obese only versus obese lipedema. What you see highlighted in red are statistically significant differences between the obese LIP group versus lean. So clearly there is an inflammation going on that is measurable in the circulation, but that doesn't really differ from people that have obese only. So then we, as I mentioned before in the experimental design, we repeated testing after the weight loss, and the data that I'm going to show now are before and after weight loss only in the lipedema group. So this came to our surprise because weight loss actually associated with a 15% reduction in both abdominal subcutaneous adipose tissue and an intra-abdominal adipose tissue volume. In addition, there was a 10 to 15% reductions in leg fat mass and thigh subcutaneous adipose tissue volume. However, the contribution from leg fat mass to total body fat mass and the android to ganoid ratio were unchanged. For the metabolic characteristics, we were also very surprised to see that women with lipedema became even more sensitive to insulin as compared to their baseline, as sensitive as a lean person could be. So despite having still a BMI that would be classified as an obese person, lipedema associates with higher insulin sensitivity. And it gets even better when there is a very moderate weight loss. And again, plasma markers of inflammations did not change much, which also came to a surprise because usually in the literature it is shown that when you have even a moderate weight loss is associated with improvement of your cytokines level in the blood, but that was not the case for the lipedema. The only change was for the CRP protein, and then leptin also was reduced. And so this is the summary of what I've just told you, that as expected that there is an increase in lower body fat in the cohort that has obesity and lipedema. However, as I mentioned, we also saw a significant accumulation of fat in the liver, meaning that also the upper body fat should be taken into consideration in these patient populations. And that insulin-stimulated rate of glucose disposal rate, which is an index of skeletal muscle insulin sensitivity, is greater in the obese LIP group as compared to the obese controls. So the next step was to look at adipose tissue biology. As I mentioned, we did we investigated how many immune cells, if you have any immune cells infiltration in the adipose tissue. And so we took biopsy from the abdomen and from the thigh within the same patients. Usually those biopsies are digested with enzymes in order to have access to the different cells that are in the adipose tissue. You have immune cells, you have endothelial cells, you have different types of cells. At the same time, we also took the blood because we wanted to kind of compare what's going on in the adipose tissue versus the blood. And so we conducted a very comprehensive analysis on the different immune cells that are usually populating adipose tissue. And so those are the list of the different immune cell populations. And then using flow cytometry, we were able to kind of get an idea of what your immune system looks like in the adipose tissue. And so we observed that there is a greater number of macrophages in the thigh of women with lipedema as compared to the abdominal, and several speakers before me have mentioned that there is a difference between M1-like versus M2-like macrophages. We are observing more M1-like macrophages in the thigh, meaning that there is more inflammation. Usually that M1-like macrophages population is involved in mounting an inflammation. However, after weight loss, there was no effect on the number of these macrophages. So meaning that despite losing weight and despite becoming more insulin sensitive, which is a good thing, your inflammation stayed the same. And then this is a graph, excuse me, this is a graph called volcano plot that relates to changes in gene expression between thigh and abdomen, and we observed almost 1,000 genes that were changed in women with lipedema. The major changes were in genes that regulate lymphatics, in genes that regulate the vasculature. The expression of those genes was reduced. Instead, expression of genes that regulates collagen, fibrosis, was upregulated, as well as inflammation. So kind of confirming what we have seen with a different technique with flow cytometry. However, when we looked at gene expression after weight loss, there was a very modest or almost no change in those genes, meaning that, again, the diet might be good for your metabolic function, for your metabolic health, but in terms of adipose tissue remodeling, we really see a very modest improvements. And so this is the summary of the study. I mean, I think it's too small to see, but just to summarize is that we were able to measure exactly where your body fat is located. And so as expected, there is higher lower body fat in the cohort, in the lipedema cohort. We do see accumulation of liver fat as well, which should be followed up. I think this is an important point. However, despite having that liver fat, women with lipedema are extremely insulin sensitive. It's unlikely that they will develop type 2 diabetes or cardiovascular abnormalities. We did not see major changes in the plasma cytokines content as compared to controls. and that levels of cytokines and chemokines did not really change after weight loss. And similar observations were confirmed for the number of immune cells located in the thigh of our patient population, and similar observations were observed for the genetic signature. So before finishing, I would like to acknowledge the people that made this study possible. So this is a partnership with the Center for Human Nutrition at Washington University. Dr. Samuel Klein is a co-investigator in this project. And also would like to thank Gordon Smith, an associate professor in the Center for Human Nutrition, who was instrumental for those studies. I also would like to thank my lab, in particular Sylvia. She is a PhD student and has a poster that summarizes some of those findings. So if you want to learn more about our project, please see her. And obviously, I would like to thank the Lipedema Foundation, who was really generous in funding our project and made those studies possible. Thank you.